genes, genetics and PCD is a really, really exciting area that's changed a lot over the past few years. And I'm really, really excited that Gabrielle has offered to talk to us about this today. I first heard her give a talk at a conference in the summer, and Katie and I were texting each other like, we have to get Gabrielle to talk to the PCD crowd. So I will let Gabrielle kick off, and I look forward to having a chat afterwards. Thank you. You should be able to share. Yeah, perfect.
Thank you very much, Lucy. Thank you for the opportunity to talk to you today. I'm really delighted. And I'd like to emphasise what Lucy said, that I'd be really happy to take questions throughout the presentation, or if you want me to clarify anything, please just ask. I'd be more than happy to do that. So I'm going to talk today about PCD and non-CF bronchiectasis in the 100,000 Genomes Project. So I will briefly talk about bronchiectasis and PCD, but you know a hell of a lot more about that than I do, so forgive my ignorance. But I will then focus on the genetics of PCD and how PCD is currently diagnosed and the role that genetics plays in that currently and can play in the future. And then I'll talk about the 100,000 Genomes Project and what we have found so far. So bronchiectasis is a condition of the airway where this part of the airway, the bronchus, becomes enlarged and inflamed and damaged. So the wall becomes damaged and you get a lot more of these goblet cells that make mucus. So you get a lot more mucus in the airway, and you also lose the cilia that normally line the airway and move that mucus up out of the airway. So it gets stuck in the airway. So it's often caused by cystic fibrosis, where you have mutations in a gene that makes this protein, which sits in the membrane of the cells in the airway and moves salts, basically. So it controls salt balance to avoid this mucus buildup. But there are lots of other things that can cause this lung damage, this bronchiectasis, and those causes vary by geographical location. So around the world, about half of causes are not known. They're idiopathic. We don't know what's causing the bronchiectasis. In the low-income countries, bronchiectasis can arise after infectious disease. In higher income countries, we have better healthcare, better provision of antibiotics, so that's lower. But you see there's all of these causes of bronchiectasis, and PCD is a cause of bronchiectasis. So in children who have bronchiectasis, up to a quarter of them will have bronchiectasis because of PCD. And I also suspect, and I think it's probably likely, that some of these idiopathic cases are also undiagnosed cases of PCD. So undoubtedly there are people in the population that have lung problems, including bronchiectasis, because they've got PCD that hasn't been picked up. And we have some evidence from that from the 100,000 Genomes Project that I will show.
Gabrielle, can I just check? An idiopathic means that there's not an explained cause for that bronchiectasis. Brilliant, thank you.
Yeah, we don't know the cause yet. So I'm not going to spend too long on talking about what is PCD. Oh, my video won't play. That's a shame. These are my cilia that I wanted to show you wafting from my nose. They were collected by Amanda and imaged by James in Southampton. But you know that PCD is a condition associated with defects in the motile cilia, and these motile cilia are found in your airway, they're found in your reproductive tract. This is the fallopian tube lining here. They're found on the tail of the sperm, and in the developing embryo they are found in a particular spot for a short time as the embryo develops, and they're there to waft things to the left-hand side of the embryo. And so if they're not doing that job, the embryo doesn't know what is left and what is right. So some people can end up with their organ placement inverted. So they have situs inversus. So the organs are the other way around to most people. So you know that the problem with the cilia in the airway leads to a buildup of mucus. People are a bit snotty, often have a wet cough. And because this mucus is hanging around, there are lots of respiratory infections, and all of that can lead to this damage, this bronchiectasis. Hearing problems as well, often. So, whereas bronchiectasis can be caused by lots of different things, PCD is a purely genetic disorder. So what that means is PCD is always caused by a particular mutation in a particular gene that has a role in making or functioning a motile cilium. So there are about 20,000 genes in your genome. There are more than 50 now that we know are associated with PCD. And they have lots of different functions. So some are involved in the process of taking these little structures which form the base of the the base of the cilia up to the top of the cell to make these multiciliated cells. So if you have a mutation in one of these genes, you have absolutely no motile cilia whatsoever. Then there are all of these genes that have different functions in different parts of the cilia. So if this is the cross section of the cilium, and these are different proteins, different structures within it, these different genes make different parts of this structure.
Sorry, Gabrielle, just to let you know, I don't know if this is the case for others, but I can't see your mouse moving if you're indicating.
Oh, if I do it here, can you see this?
Yeah, now I can, yes.
Oh, right, I'm on the wrong screen.
No, don't worry.
Okay, so mutations in these ones mean you have no cilia. Mutations in some of these ones mean you can have a problem with the structure of the cilium, so it looks weird when you cut it through, or they don't beat whatsoever, they don't move, or they move in a funny way, or they move too fast, or they move too slow. So there can be lots of different problems with the cilia, but the cause is always a mutation in a gene that is responsible for part of this structure or function. So we have a pretty good understanding of the genes that cause PCD. So we know that there are more than 50, but that doesn't give a genetic diagnosis for everybody with PCD. If you took 100 people with PCD and you looked at all of these genes in all of those people, about 70 people would get a genetic diagnosis. So we'd find the mutation that's causing their disease in one of these genes, and the other 30 wouldn't. And there are a lot of reasons for that. So there could be other genes in the genome that we don't know about that are involved in this process, which when mutated can cause PCD. But there could also be mutations in these known genes that we're not picking up, because there's a massive challenge in genome sequencing in genomics that I'll cover more in a minute, which is trying to work out which of the variants in your genome are the ones that are causing disease, because we actually have a lot of mutations that we're carrying around, but most of them don't cause problems. So how do we diagnose PCD currently? It's using a suite of tests that involve measuring nitric oxide in the nose. You will know this if you've had your PCD diagnostic tests. There's high-speed video imaging of the cilia to look at the beating. There's imaging of the cross-section of the cilium using what's called transmission electron microscopy. So this is trying to pick out these very specific changes that we see, and that can actually help us to work out which gene has gone wrong. And we can also put fluorescent tags on the proteins that are found in the cilium, and then we can look at those under a fluorescence microscope to see, are they lost from the cilium. So here, for example, this is some healthy cilia, and these proteins are here in green. This is the cilia from a patient that has a mutation in this gene, and you can see that it's lost. So it's based on clinical features and clinical history and these tests. And genetics is often used, but it's not a sort of official part of the diagnostic pathway. But there's real potential for improving diagnosis using genetics, because it's PCD is a genetic disease, And so if you can find the genetic cause, you can give, really quite a confident diagnosis. And this can be useful because, as I mentioned, in different people, the cilia beat change can vary. The structure can vary as well. So some people can have PCD, but the structure of the cilia looks normal. Or some people can have PCD and actually the motile cilium defect is really, really subtle and difficult to pick up. So actually you could have PCD, but some of these imaging analyses might miss it. And there are definitely cases of PCD that are missed because we don't have a perfect system for detecting it. And I think genetics can play a really helpful role in catching some of those cases. So historically in the UK, if you wanted to have a genetic test for PCD, you would typically go to one of the diagnostic centres in Southampton or Leicester or London, now also Leeds as well, of course, and they might send a sample of your DNA to one of the local labs for testing, where they would test some of the known PCD genes. You'll see that these numbers vary, and that's because over time we find more and more genes that we know cause PCD. And one of the limits of this approach of just looking at the known genes is that we're picking up new genes all the time, and we don't always keep up in updating the genes that we're looking at in patients. You could also have a test in Exeter that looks at all of the genes that have ever been associated with any human disease ever, and that's called the clinical exome. But you'll see that there's a very there's potential for real disparity here in access to genetic testing. So not so much in PCD actually, but in other rare genetic conditions, there was a real difference in the access to genetic testing that people had available. So if you lived near a large centre with a large genetic testing centre, you would be more likely to be sent for genetic testing if you were suspected to have a genetic condition. And so there was real inequality in access to this kind of diagnosis around the UK. So quite a number of years ago now, 8 or 9 years ago, the NHS and the government, the UK government, developed a vision to overhaul the way that genetic testing was carried out in the UK, to introduce a truly national genomic medicine service. And this would involve not just sequencing of a few genes involved in different diseases, depending on where you lived in the country, but national access to, in some cases, complete sequencing of your whole genome to try to get a diagnosis for rare genetic disease. And so, whereas in the past, you would go and see a clinician or a consultant, and they would need to know the pathway to genetic testing, and they would need to know who to contact to order that genetic test. The idea with this is all of the tests are arranged in a national genomic test directory. There's a website, any clinician can go in and book that for their patient. So it was a really, really good initiative with honourable intentions, I think, to improve equity of access to this element of healthcare. So historically, a lot of PCD genetic testing was done in Wessex, which is our local lab in Salisbury, close to Southampton. And Wessex and Birmingham have teamed up to form one of these lab hubs, and they are still the group offering PCD testing. I think the current test is now 46 genes, but that will be updated every year, I think. Okay, so how do we actually set about introducing this National Genome Medicine Service? This involves a complete overhaul of the way we do genetic testing in the NHS. And the 100,000 Genomes Project was the way that this was implemented. So it was a hybrid clinical diagnostic and research study which aimed to sequence 100,000 genomes from 70,000 patients and their family members. So you'll notice that there's a difference there. How do you get more than 70,000 genomes from 70,000 people? Well, it included a lot of patients with rare genetic disease like PCD, but also patients with cancer as well. So cancer is not an inherited genetic disease, but an acquired genetic disease. So in your lifetime, you acquire mutation that causes your cells to become a tumour. So that tumour has a different genome from the rest of your body. So in those patients, you would take DNA from the healthy tissue and the tumour, so you get two different genomes to compare. And this produced 21 petabytes of data. And to put that into perspective, 1 petabyte of music would take you 2,000 years to play on an MP3 player. So an awful lot of data. So one of the big challenges was data storage and secure data storage. So that was a lot of the early work of the project. So it was organised right across the country, 13 genome medicine centres, 85 NHS trusts, 1,500 staff, and I would think more than 2,500 researchers around the world. So to understand genomics, we need to understand genes. So I'm going to take you back to basics. I hope that you all have heard of DNA and be familiar with the fact that it's a double helix structure. And that double helix is made up of 2 strands. So here we've got 1 strand, and we have this backbone, which has really strong bonds holding it together. And I've put these 2 things at the ends to show that it has a direction. So the 2 ends are different, and we call them the 5' and the 3', and you always read it in a particular direction. And so that's really useful because it allows you to store information, and that information is read in a very specific way. And the information is held in these bases. So along the backbone, you have these bases. There's 4 different ones, A, C, G, and T, and they form a sequence to hold lots and lots of information. So that's 1 strand. The other strand pairs up, always A to T and always C to G, so that you get these 2 strands pairing up. You'll notice that this one goes in the opposite direction, so we call them anti-parallel, and they're complementary, and it stores all of this information with 4 letters, and the bonds between the 2 strands are actually really weak, so they can be prized apart really easily, so this information can be accessed really easily. So across your whole genome, you have 3.2 billion bases, and that's not one single long piece of DNA. That's across 23 long pieces of DNA that come in pairs. And we call these long pieces of DNA chromosomes. You get 1 from your mum and 1 from your dad. And these are the 23 different pairs. They're numbered from 1 to 22 in order of size. And then we have the sex chromosomes. So females have 2 X chromosomes, males have an X and a Y. All of the chromosomes have genes on them. But the genes aren't easily spaced along them. And there's also a lot of DNA which is not genes. Okay, this is something that a lot of people aren't familiar with. Your genome is all of your DNA in your cell, but actually the majority of that DNA is not genes. So genes are things that make proteins, you know, like those building blocks of the cilia. But actually about 98% of your genome doesn't make protein. It has other functions, regulatory functions, structural functions, functions that we have absolutely no idea about. But the really critically important bits are the bits that make the proteins, for example, the bits that make your cilia. So they're found across all of these chromosomes. If you spread them out end to end, it's about 2 meters. which is about the height of a forklift truck. So if you look at these chromosomes, these really, really dark bits are bits where it's really, really highly condensed. So obviously you need to fit 2 meters of DNA into one single cell, tiny, tiny cell. So your DNA is wrapped around proteins and it's folded in certain ways. So it's really, really, really tightly packed in. Amazing, amazing molecule. So if you wanted to read your genome letter by letter, and you read a letter a second for 24 hours a day, it would take you 100 years. All of that information in every single cell. And it's incredible how stable it is. If you think how we have evolved over tens of thousands of years, all of that has been by DNA. So DNA is incredibly stable over generations and generations and generations, but changes occur. Okay, so we have variations within our genome from person to person. Here's a variant from this piece of DNA to this one. This would be a sequence variant, a small sequence variant. Here's another one. You've got a small base deletion here. And there are about 4.5 million in each person from individual to individual. And they account for normal variation, hair color, eye color, height, elements of your personality. Obviously, a lot of these interact with your environment. They're not purely genetic. But then also some of these variants contribute to disease. So if you inherit them from your parents, they will cause inherited genetic disease. If you acquire mutations, for example, through UV exposure, in ionizing radiation, whatever, then you can develop cancers. They're acquired genetic conditions. Oh, I also want to mention that you can also have big deletions within your genome as well. Some of them completely fine, they don't cause any effect. Some of them can cause disease. So that's important because we'll come to that in a minute. So we used to be able to sequence DNA about 300 letters at a time, 300 to 1,000. So we used to have this technology developed by a guy called Fred Sanger. He won the Nobel Prize. He actually won 2 Nobel Prizes. Really smart guy, and apparently really nice as well. So he developed a technology which basically involved sticking fluorescent tags on those 4 letters. The 4 letters have different colors, and then you read along the molecule of DNA, and whatever color you see, you work out, okay, well, that's that letter, and you've got the sequence. But you could do that 300 or 1,000 bases at a time. And it was that technology that was used to sequence the first human genome, which I'll remind you, the genome's 3.2 billion letters. So obviously that took a long time, start to finish about 13 years, and cost £2 billion. And this new technology came along 2008. So this was about halfway through me doing my PhD. Completely revolutionized everything. And this technology uses the same principle. You stick a fluorescent tag on each letter, but instead of looking at one molecule at a time, you have what's called an array of millions of short pieces of DNA that you sequence all in one go. And you have a very clever camera that takes photos, a series of photos, and then it works out the sequence of all of these different bits. So with this technology, we can now sequence a whole genome in actually less than a day and much less than £1,000. I need to update that. So with that new technology, we've been able to really, really rapidly increase our understanding of genetic disease, and also use this technology for diagnosis. So it was announced officially in 2014 that the 100,000 Genomes Project was going to be going ahead. There was a pilot study before this. And the aim was to bring in whole genome testing of patients to try to understand the cause of their diseases. So what did this involve? In the case of rare disease patients, the DNA sample was taken from the affected individual, and the first person that you study who's affected you call the proband, if you've ever heard that term. So this would be the proband, and then both of their parents and sometimes other members of the family as well. And in the case of cancer, some healthy tissue from the person and their tumour. Why are we interested in the parents? So you'll know that in the vast majority of cases, PCD is a recessive disease. So you know that you have 2 of each chromosome. All of your chromosomes are in pairs, and you get 1 from your mum and 1 from your dad. So in this pair of chromosomes, there's a copy of each gene on each chromosome. In the case of some diseases, you only need to lose 1 copy to get a disease. But in the case of PCD, you need to have a mutation on both copies to actually get the disease. And so in order to try to work out what's causing the disease, it's really useful to know what's come from mum and what's come from dad. So if you find 2 mutations in a gene in this person, and one's come from mum and one's come from dad, that's more likely to be causing their disease. Whereas if they both came from mum, that can't be the cause. Okay, so patients were generally included if they'd had the previous genetic tests already done. So they'd had the known, for example, PCD genes tested and nothing had been found. Then they were eligible to go on and have their whole genome sequenced. Towards the end of the project, they wanted to just sequence anybody. So there were some people that went in who hadn't had previous genes tested. And then there was a long consent conversation, because this genome data was going to be made available to researchers, and they wanted to know if people consented to that, if they wanted to get other findings. DNA was taken and stored in a central biorepository, and then all of that clever sequencing was done, and the initial analysis was done by a company called Genomics England, which is actually owned by the Department of Health. It's a government body. So it was all automated. And then those findings were reported back to the centre where the patient was recruited, and then they should have been fed back to the patient. That is what should have happened, but I know that it didn't always go that smoothly, and I think often at the beginning when people were recruited, they were also given unrealistic expectations of the time that it would take for these results to come back. So we are 6 or 7 years down the line from the start of this project, and I believe that most, if not all, results have been fed back now, but I'm not even sure. So those results were fed back to the centre where the patient was recruited, and then that centre had to do their own analysis. They had to look at those findings and decide, do they think that that's the cause of the disease? And they do that using some guidelines from the American College of Medical Genetics. And there's a series of criteria, and you look at this genetic variant and you look at the information from this patient, and you have levels of evidence to decide whether that genetic change is likely pathogenic, so likely to cause disease, pathogenic, definitely causing disease, unknown significance, or benign, so it's not causing the disease. And then for everybody where a clear result wasn't found, there was the research arm of the study. And people could elect to receive the main findings. They could also elect to get additional findings. So there are, if we're looking at your whole genome, we might find something we don't expect. So there are genes which, if you have a mutation in them, can predispose you highly to things like breast cancer. So people could opt in or out of getting those additional findings. It was only things that were actually actionable. So, you know, if you got this information, something could be done about that clinically. So I believe most people opted in to receive those. And you could also then find out if you were a carrier of other disease genes as well. So this was the timeline. So yes, it was initially announced 2012 and the pilot, and then it was officially announced 2014, and they set up these genome medicine centres around the country. First diagnosis came in 2016, but the recruitment wasn't complete until 2018, and a lot of the results weren't reported back until 2019 and 2020. And the Genome Medicine Service has been sort of kicking in from April last year, although COVID kind of got in the way of that a little bit. So these have overlapped a bit weirdly for some reason. But this is just to show that of all of the patients that were recruited to the rare disease arm of the project, you can't read it. This big chunk is neurodevelopmental conditions. Then there was a big chunk of cardiovascular conditions. This, I can't read actually, possibly renal, ophthalmic conditions, ultra-rare disorders, tumour syndromes, and then ciliopathies was about 1% of all the patients recruited. So 274 people with PCD, or sorry, 274 people in the PCD category, so that's 118 people with PCD, and then the other individuals are family members, either unaffected or affected family members. So a lot of people were involved in this. And then there were 142 people with non-CF bronchiectasis, and including their family members, 249 people in total. And sorry, this is formatted strangely. Maybe not surprisingly, most of these were recruited near the diagnostic centres. So Wessex, which is Southampton and Portsmouth, North Thames, which was Brompton. This is East of England, which covers Leicester. And then there was Greater Manchester, West London, and other places. But most of the cases of non-CF bronchiectasis were recruited in Wessex, in Southampton and Portsmouth. So that's how people were recruited. Their DNA was taken and sent for sequencing. And then once we've got their sequence back, we can compare it to a reference genome, so a standard genome from an average person, and comparing it to that, we pick out all of the variants that are different to that reference genome. So in the average person, you'd have 4.5 million variants from the reference genome. How do you get from that number of variants to work out which is the one that's causing their disease? Well, the first thing you do is you get rid of anything that's common. So a lot of these things will be common between lots of different people. They don't really have an effect. Lots of people walking around with it, it can't be causing disease. So that reduces it a lot. If there's only one variant in the gene, so only one gene is affected, and we're looking at a disease where both genes need to be affected to cause the disease, get rid of those. And then we've only got rare variants where both genes are affected on both chromosomes, okay? And then we can go and look at mum and dad, and if both of those variants are in mum, we can chuck those out. If both of the variants are in dad, we can chuck those out, because we know that one has come from mum and one has come from dad to cause this inherited disease. And that leaves us with more of a manageable number of variants that then we can look at, but still quite a number. So how do we prioritize the ones that we look at? So we focus on variants in the genes that we already know cause disease. And so for the analysis for this project, they used this panel of genes. So although there are more than 50 known PCD genes, there are some where not many cases have been reported, so the confidence that it might be found in other people is lower. So they had these 32 genes that they were confident, absolutely confident, cause PCD. And so they were the genes that were looked at first. In non-CF bronchiectasis, in most people, they looked at the PCD genes, but not in all people. So some things were missed there. And then they also looked at CFTR and these other genes that are known to cause CF. And they also looked at all of the genes that can cause primary immunodeficiency, because that can be a cause of non-CF bronchiectasis. And then these results are fed back to the recruiting centre. And if they have a mutation which has a high impact on the protein that that gene makes, so the protein's truncated or lost completely, we'd call that high impact. But then there can also be mutations that just very, very subtly change the protein. And so we'd call that a moderate impact mutation. So that would be lower tier. We'd have lower confidence that actually that is likely to cause disease. And then there were also variants which were rare that affected proteins that weren't on those gene panels. So if we look at the results that Genomics England analysis came up with from 125 affected individuals with PCD, there were confident diagnoses for 39 people, so about 31% of cases solved. And then there's a possible diagnosis, so some of those moderate mutations in another 21 people, so they could potentially get diagnoses. And it's in all of these different genes. So DNAH5 and DNAH11 are common causes of PCD in the Caucasian population, partly because they're just massive genes. So genes vary in size. These two are absolutely massive, so they're just more likely to get a mutation. But CCDC 39 and 40 are common in the British Asian population. And then you see all of these other slightly rarer causes as well, although DNAI1 is not uncommon. In the case of non-CF bronchiectasis, if we look at the Genomics England analysis, of 143 people, actually only 7 have mutations that will give them a confident result. So a high impact mutation in a gene that has been known to be associated with non-CF bronchiectasis. So that's only about 5% of those people that went in. And most of those are cases of PCD. And then there's another 8 that might have mutations that are causing their disease, but we need a bit more information before we can be really confident about that. So these are possible diagnoses, but we need some more information. And so that would be another 5% of people. And again, most of these are PCD genes. So we've got a good diagnostic rate in PCD, at least 31.2%, possibly up to 60% if we can prove that those maybes are definites. But in non-CF bronchiectasis, much lower, okay, 5 to 10%. And nearly all of those are cases of PCD that haven't been picked up. So this is an important point. What happens to all of those other people that don't get diagnoses? Well, that is then the job of the clinical interpretation partnerships with these researchers around the world. So you have to apply to join one of these to get access to this data. You have to prove your credentials. Not anybody can access this data. You have to prove that you have the expertise to look at this, and you have to be associated with an organization, an institution like a university who can vouch that you're legit, I suppose. So I've got lots of people to thank, and I've probably forgotten some people. But it's important to say that this is very, very, very collaborative work that's involved a lot of people. Obviously, Jane and Wolf and Mary and all of the diagnostic team in Southampton, the people in clinical genetics, in the genetic testing lab, and Genomics England.
Thank you so much. I didn't want to stop you because it was so interesting. We've had some really, really good questions come through. I think this is it's always a bit of a, I think if you're don't have a scientific background and you have this condition, it's a little bit of a maze to really understand, you know, what it means and kind of the difference between like genes, mutations, and so that was really clear. If you do have further questions, do drop me an email, and we'll do our best to, maybe if Gabrielle doesn't mind, if there's any follow-up questions.
Oh, you know, I'd be delighted.
Thank you so much. I just want to say thank you, because I realise you're really busy at the moment. Gabrielle has had a huge discovery on the COVID front in her research, and is very busy with some COVID research at the moment that went into a very prestigious journal the other day. So that's amazing news, and I realise that has meant that you're super swamped with work at the moment. So thank you so much. It's been really, really helpful, and yeah, really look forward to kind of carrying on these discussions, and hopefully in 5 years' time we'll have another talk and you'll say, oh, we've discovered all the genes, it's complicated, you know exactly how it's caused.
And then I'll have no job.
I won't.
There's always something to
yeah, I'm not sure what to wish for there. Thanks ever so much. It's really, really lovely to see everyone, and I hope you guys are all doing well and staying relatively sane in what is a endless lockdown and shielding. But the vaccines are coming, and gradually it is looking better, isn't it? So lovely to see you all. Take care. Thank you ever so much, Gabrielle, and we will see you again soon for the next one.